talk to you about a body of work that has taken me basically since I was a postdoc with Karina and Vladimir at Nebraska. So at the time, we were trying to use these topological ideas just to see if there was structure in neural activity in general. That was sort of our big goal. And I think we can claim to have succeeded at that. And when we took this out and showed it to neuroscientists, they said, "That's great. What is it for?" And we said, "That's a great question. We'll get back to you." So I've spent a lot of time since then thinking about two things. One is why is this tool so successful at seeing structure in neural activity? And then what kinds of things can we use it to really do? And a lot of that has taken me in uh, sort of interesting directions where we had to develop some new mathematics in order to develop to really understand how to use these things. This talk is going to be light on details and heavy on ideas. And then Nico, who is my postdoc when I was at Delaware, who is now at Rockefeller University, is going to take over in about an hour and a half, and he's going to show us about an hour, and he's going to show us some of the how, some of the details that I'm going to brutally skim over. All right. And before I get going, I just want to say that I was involved in this work, but there were a lot of people. So Iris Yoon, that's wrong. She is now at Swarthmore as of this fall, wonderfully. And Nico were both postdocs who worked on this work with me. Rob Greist at Penn is one of the founders of applied topology, and so he was a great mentor in all of this. And then we have some experimental collaborators at UCSD, Spencer Smith in his lab. And then there are more people that I can name that are responsible for this work. So, okay. So I want to start with something that should be familiar to everybody in neuroscience, which is receptive fields, right? This is just a sort of a foundational piece of lore at this point, right? So these are, hopefully familiar, right? I have these simple cells in V1 in primary visual cortex, and they are sensitive to position and also orientation of stimuli, right? So they're edge detectors on the retina. And we really understand really well how that comes from pooling neurons on the retina, right? This is a well understood story. But you do some work, right, on data, and you get pictures that look a little bit like this. So these are what each neuron sees represented in some mathematical way. And there's a nice model you can put on neurons when you have this sort of receptive field that you've imputed, which is that I can model the rate of firing of a neuron, right? So the rate of the ith neuron given a stimulus is some rectifying nonlinearity applied to a pairing between the stimulus and this particular function, right? And the pairing can be as simple as just a convolution or whatever you feel like putting in there, depending on your choice of model. I don't want to be light on the details today. But right, if we do this in computational neuroscience, this is how we use these. Yes. So sorry. So you're not allowed to have a negative rate. Right, and I don't want to compute angles. Yeah, so thank you. So that's something like, if you like, that's just 0 if you're less than 0, and identity if you're positive, which will come up later this week, I'm sure. But choose your favorite. Okay, so anyway, this is just a very standard model, and the thing about that model is that we look at those pictures and we can't help but notice that some of them are more similar to one another than others, and so it's very tempting to start assigning distances between them, right? It feels like the right thing to do, right? Those two, F1 and F2, those look really, really similar. They're probably really close to each other in some way. Whereas like 1 and 3, not so much, right? And there are lots of ways you can do this. Again, here's just, a way that I picked out of a hat, which is just the L2 norm on functions, right? But you can choose your favorite. Mostly what I want to do is think about those pictures up there as vectors in some space, and then I want to, put a metric on it. But I've only got finitely many of these things, right? Because I've got finitely many neurons I'm observing. And so the resulting object is going to be a finite metric space, right? It's just a bunch of pairwise distances. Okay. And so I start with that, and then I think to myself, well, look, I've got these pictures. I could imagine that there's another neuron I didn't add. It should give me a similar picture. So really underlying this thing is some continuous big metric space that these things are all just sitting in. And I should be thinking about everything that way. It's a very tempting thing to do. And neuroscientists, right, have dubbed these things neural manifolds. And I take mild issue with that name. I think I'm going to call them stimulus spaces today, but after we leave this room, we can go back to calling them neural manifolds. And we're talking to people who use that terminology, but the word manifold has a meaning. I like that meaning to be what it is. And okay, so we're going to say stimulus space today, but you can, if you prefer, you can think neural manifold. Okay. So this is what I want to do, right? I want to, yes. Yes. So the F's are F's are just functions on the stimulus. So I've got like the retina is just a rectangle in this picture, right? And so I'm integrating or I'm convolving that function I've listed there where blues are positives and reds are negative or something like that. It's just sort of schematic, right? But I'm going to convolve that against whatever is being presented to me, right? And I'm going to get responses. Well, I don't even convolve, just pair. Imagine that what I'm getting is a bump function. I just want to pair that bump function on the retina against that thing and see how much of a response I get. Don't worry too much about the technical details, but yeah. Right. So I claim that if you do this, that's what you're doing. Right? Because the brain doesn't ever have this. It has a finite number of neurons and they represent some things. And then I come along and put this logical layer of the neural manifold on it, coordinatizing my neurons. Right? So I mean, we can argue about, whether or not I'm using the neuroscience version of neural manifold entirely properly, but I think this is, in some sense, what people are doing when they say there's neural manifold, right? They're saying, okay, I want to take all of my population activity and put coordinates on it. Well, I claim that if the stimulus space is there, then yes. Yeah, right. These two things should align.
I would like to think of the input space as a stimulus space where the stimuli, right? And the output space is something within the, that you can think of as embedded in the space of neural activity.
Okay.
The output is maybe what people call neural.
Sure. And so I claim that I will agree that there is some questionable alignment of terminology here. I'm trying to simplify that side of the story just a little bit. So let me just go back one slide. So I think the F's here. So sorry. Let me go back. You can do it. Oh, it's thinking about it. So the F's are, right, so this is the neural activity, right? So this is the thing that you're thinking of as making up the neural manifold. Yep, right. Here, this is the thing that this is the receptive field for the individual neuron. Those are the things that carry geometry, right? Because that's what I'm trying to construct the geometry out of is those things, right? That's why I think there should be geometry. Right. So I've constructed a behavioral or stimulus correlate. And these things then are responses to that correlate, right? There's some way of measuring response to that correlate. So yes, that's right. Yeah, S and Fi are both functions on a square. Although one of them I might think of as sort of a dual object. Yeah, and it's intrinsic in the fact that I put this pairing in here, right? So there's an isomorphism there that I don't want to think about too hard. Okay, so but I want to claim to you that so if you'll follow me for just a little bit, I think I can get you to a place where you'll agree with me. At least maybe under one model. And this may be non-standard terminology. I tend to exist in a vacuum way out in Oregon. I think about things weirdly. Okay, so here's some well-known examples. So here's the one we were just talking about, right? So simple cells in V1, right? Their stimulus space is something like position on the retina, but also angle, right? So those are coordinates that I'm putting on these cells. And I'm thinking that there's this underlying continuous object which coordinatizes these things. All right. Place cells, right? These are very well known, simple things, right? Cells that correspond to locations in the environment. And then there's these ones over here, and I am required by law, if I'm giving a topology neuroscience talk, to bring up grid cells, because you look at the picture of the receptive field of a grid cell and you say, ah, look, that's a whole bunch of points sitting on some kind of hexagonal lattice. There's nothing interesting going on there in terms of localization in some space. But then, some topologists come along. They say, look, I've got a hexagonal lattice and I've got a localized location in that hexagonal lattice. I can just look at one of the hexagons, right? And I was told that many of you might not be topologists. So you might not think what I'll do is I'll just take, right? If I'm on this hexagonal lattice and there's symmetry, I can go off one edge and come back on the other edge. So these are gluing marks, right? I glue the 1 arrows and the 2 arrows and the 3 arrows together. So I just imagine, right, that I'm going to be insensitive to that in terms of my response. And then so now I just told you I'm gluing, right? I imagine going off one end and come back in the other end. So I'll just actually do that gluing. So let me glue those 1 edges together. Now I've got something that looks a little bit like a funny cylinder. And now I want to glue the 2s and the 3s together. And in 3D, I have to kind of grab and twist and glue. But what I end up with is a localized point on a torus. So, right, and there's very, very nice, very nice work from folks over in Norway at NTNU, Moser Lab, and some associated mathematicians who demonstrate that, in fact, you can always look at activity in entorhinal cortex, grid cell activity, and find this thing. It's in there. So that's very nice. So this is position on a torus. Okay. So these are the kinds of spaces that I'm expecting to find, and maybe more, right? Like you might think that there are much more intricate things going on in higher visual areas than just these simple geometries. And I personally think that, but it's hard to get the data to prove it. Okay. So here's what I want to do. And this is, what I claim that we worked on in Nebraska all that time ago, right? If you have population activity, I want to know whether there is a stimulus space hiding underneath. I just want to look at neurons acting, and I want to say, is that thing in there, right? Is there this geometry? And if so, I want to know what geometry am I looking at? And the important thing here is I don't want you to cheat and tell me what is being represented. That's right, really, really vital to this whole enterprise. If you're allowed to tell me what the neural network is doing, then I'm just going to write down what the stimulus space ought to be by knowing the geometry, right, of the stimuli. I don't have to know anything else. So this is the big challenge that we want to take on, because I want to think about brain regions, right? You need to win a Nobel Prize in order to figure out what these things are, it seems like, right? We've got Nobel Prizes for grid cells, Nobel Prize for place cells, right? So I don't want to have to do that level of difficult work in order to identify what the geometry intrinsic is. I just want a method that's going to do it for me. So, okay, so you might think, and many people do, that the right thing to do now is to visualize your activity, right? I'm just gonna, maybe I can see this stuff if I just visualize it. And this is not a bad starting point. So this is a picture so these are some head direction cells in a freely behaving mouse, I believe. And so those are the decoded receptive fields for the head direction cells up there. And you can see that some of them have sort of nice unimodal behavior and some of them don't. This is just the kind of thing that you're likely to see if you're looking inside the brain of an animal and you don't have the ability, right? But I'm going to pretend I don't know that information up there. So I just have the activity of these neurons. So I'll build a correlation matrix. And now I just want to, visualize the data in that correlation matrix, right? How similar are these things? If that's a circle, if that's head direction, then I should be able to do a circle. And it turns out that whichever of your favorite methods you try, sometimes you succeed, but more often than not, it just doesn't work without a lot of pre-processing based on knowing already what the answer was supposed to be. And this, people look at this and they say, well, you see the circle around the outside there, but that's actually just an artifact of the multidimensional scaling algorithm. It just pushes everything onto a disk. There's no circle here, okay? And if you want to do a torus, you're even further out of luck. So just visualizing things might get you places, and neuroscientists have been very clever about visualizing things and finding ways to do this, but usually you precondition on knowing what you're looking for. And if you have no idea what the shape is going to be, you need something more principled. So I am here to claim to you that topology is that thing. So what I want to convince you is that if you're looking for this kind of geometry and structure in neural data, you want to start from the bottom up. So okay, so now here is where Nikki and Karina and I are going to argue about what stimulus spaces are. But here's my model of a stimulus space, and you are certainly welcome to tell me that this is the wrong usage of that terminology. Like I said, living out in the middle of nowhere. So Oregon's not the middle of nowhere, it's great. But it certainly is far separate from a lot of the places out here east. So, okay, so I'm gonna say that a population of neurons has a stimulus space coding scheme if there is some metric space, which I'm going to call the stimulus space. And I'm going to embed the neurons in that stimulus space as a collection of points. So they're just going to live in there somewhere. Okay. And now I'm going to model the firing rate of each neuron in response to a stimulus, which is going to be a point in that space, simply as a monotone decreasing function of the distance between my point, my stimulus, and the locations the neurons are corresponding to. Okay. And so this is me just trying to sort of put a little cover over the fact there's a difference between what the neurons are doing and what the right, and what the stimulus is. All right. And now I'm going to define the receptive field of a neuron at some threshold theta, just to be the sort of super level set in the space of that rate function. So I'm going to say it's everywhere that the rate is above a certain amount. And if I have a monotone decreasing function, then what happens is I just kind of have a little ball around each one of these points in which that neuron is super threshold, in which it's on. Okay. So this is my model for neural activity. I want this to be the thing that I'm trying to find. Right? Absolutely, yeah. You can. So I'm leaving it simple in this on this slide, but there's absolutely no reason why those apps can't be different. However, I claim you could probably, well, I have to think about this very carefully, probably incorporate that into the metric if you were feeling saucy.
I'm just thinking in the grid cell example, right? You have grid fields with different types. So you have many neurons that might have exactly the same
Yes.
position. Right?
Yeah.
But they will be different. They will have different.
Yeah, absolutely. And yeah, you're absolutely right. So that's the I always leave the FI off of this particular slide, but there are a great many situations where that's an intrinsic feature. But it really doesn't make a ton of difference. Yeah. Sure. Yeah, absolutely, absolutely. So yeah, a lot of different ways to write this down, and the kernel approach is useful in a lot of ways. In particular, I think it's more technically correct. Thank you. Well, let me ask you. So let me tell you. I will tell you no later. But for right now, I'm just that's like models as simple as possible in order for it to be useful. But let me also say that if you have non-unimodality, there's a question I have, an open question that I have. then the question is it because your receptive field was not unimodal or because you drew the wrong picture, right? And maybe this is the right picture. So I don't know. Okay, but let's, you'll let me use this one for a little while and we'll come back and fix it during the workshop. Maybe people can, okay, so one thing I want to observe is that if I do this, if I have enough points, if I have enough neurons scattered around this thing, and I assume some very mild technical hypotheses, then I'm going to, then this neural population activity is actually going to embed this thing in neural activation space. And that's simply because I'm going to have enough different functions on this manifold or metric space to give you a nice embedding. Okay. And so that's why I think we can pretend that the neural manifold and the stimulus space are the same. Okay. Because I think that one of these is just an embedding of the other. Or in any mathematically nice world, it would be. Now, of course, there's a whole lot of things you have to take into account when you're thinking about real neurons, including noise and yada, 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 yada. But I'm trying to make for you a very nice, simple model. Okay. Okay, I want to show you a couple of videos. A couple of you might have seen these videos before, but I think they're really interesting. So here, the idea is that I have just a circle, and I've got two receptive fields, and they're very dumb little triangular bump function receptive fields. So I've got two neurons there, neuron 1 and neuron 2. And what I'm going to do is I'm going to run a stimulus just walking along that circle, okay? And then I'm going to show you the embedding. So this is the input neuron response and the output neuron response. So the input neurons are just those two neurons, and the output neuron is some of those two. And I just want to show you, actually pay attention to the input neuron response, because that's actually the thing I'm interested in showing you here. So these two receptive fields, right? So this is neuron 1 axis and neuron 2 axis. Or I think, yeah, neuron 1 axis and neuron 2 axis. Let's see if this thing will actually play on this laptop. There we go. Okay, so you can see, right? I get a response from neuron 1 and a response from neuron 2. And in between, I have sort of this joint response where one is decreasing and the other is increasing. Okay, so I claim that's a chunk of a circle embedded in neural activity space. Okay. And it's just this trace of this thing. And of course, if there were many neurons, then it wouldn't do this axis aligned thing. It would be moving along, but it'd be a sort of angular trip. If I have the two neurons, if the receptive fields overlap a little bit more, you get this other interesting phenomenon that happens where now you get something that looks a little bit more like that. So you get interesting phenomena even with these silly little receptive fields on a silly little space as I sort of sweep through. Yep, absolutely. So I'm coming right to left here, right? And you'll see that neuron 1 activates, and then neuron 1 and neuron 2 are both coactive almost immediately. Right, so this is neuron 1 activity and neuron 2 activity. Right, so I come here, and then I'll go up here, and then at some point neuron 2 will still be growing in activity, but neuron 1 is falling off. Right, and so I get this sort of other funny behavior, right? So there's some interesting geometry to what's happening here in this embedding. Yeah, so I'm just, yeah, time is sort of tracing out like that. I'll run it again. On the right, yeah, that's right. So. That's right. Yep, that's right. Yes. So the claim is I'm embedding that little chunk of circle in neural activity space that way. That's the sum of these two, and that's just part of this video for we were doing an experiment, and I dragged it onto the slide. But the top is really the important stuff. So anyway, it's just there's this interesting geometry on the embedding, right? It's not just a trivial like there's a circle here. Stuff is happening, and so I claim that it's not immediately obvious what's going on. And one question I have in general for the neuroscience universe is just this: what's that embedding for? right? The brain could have chosen to do this in a lot of different ways. There's a geometry to this embedding. It's an interesting geometry. It's not the one we think about in mathematics normally, which is how small can I make the embedding space? This is sort of how big can I make the embedding space, or how, it's all locally embedded in some way. And so I think there's a lot of interesting questions mathematically, geometrically about this approach that the brain takes to representing these things. And one thing might be learning, and we'll see that if I ever get there, which I might not because I'm taking my sweet time. So, but I can talk to you about it offline later if I don't. All right, anyway, so, okay, so, can I go forward? Probably not. No, I'm stuck in some weird mode. So, right, so one thing that I want to observe here is that this neural coactivation thing, right, is not just a complete nothing. It tells me that if I have overlapping receptive fields, then I should observe more neural coactivation. And that's not too shocking, right? This should be a deeply non-shocking observation, but I'm building up instead of down. So I'm trying to construct a version of this story where I'm going to tell you what the answer should be when we look in the brain, not the other way around. Okay. So why is topology a natural thing to do with that stuff in mind? Okay, I've got this little model that I built, which we can maybe change the terminology on later. But suppose I have this stimulus space and a collection of receptive fields that cover it. So I've got enough neurons to at least cover this thing. And here, I'm just going to imagine it's a circle. So there's my receptive fields. Okay, they cover it in some kind of funny way. And I'm a mathematician, so I need a small enough example I can draw. So if you are not a topologist, you may not know about this fancy construction, but this is, in my mind, this is the most important theorem in topology in some sense. I'll probably get my topologist license taken away from saying that. But if you give me that collection of sets and the intersections of that set, I can construct a new object, a combinatorial object out of that intersection data, which we call the nerve for historical reasons, which has nothing to do with neuroscience. So how am I going to do it? For each one of those sets in the cover, each one of the receptive fields, I'm going to build a vertex. Okay, so I've got one vertex corresponding to each one of those things. And now I'm just going to record the intersection data by putting in faces. If you haven't seen simplicial complexes, they're just like graphs, but you're allowed to have 7 or 8 or 22 things involved in what's called a face, not an edge. But you have this downward closure condition, which says that if I have a face, I've got all of its subfaces. So I can put an edge for each pairwise intersection. I can put a triangle in for each 3-wise intersection. I don't have any 4-wise in this picture, but you imagine I put a tetrahedron in, and if I have 17, I put in something that 17 simplex, or 16 simplex technically by dimension. Okay, so I'm going to build this object. And the thing you might observe if you squint at it real hard is it looks a little bit like a circle, right? And you might hope that that's not a coincidence. There is this, so the top is the rough version and the bottom is the actual theorem, said without as much notation as I would like. So if this cover is good in some way, which means technically if the intersections, all the intersections are either empty or contractible, meaning they can be collapsed to a point, then this thing I built is the same shape mathematically, topologically, as the top one. And there, I mean, continuous deformation. So you imagine I can stretch and pull as long as I don't glue distant points or cut things. Okay. And you can see, probably just glance at that thing and see how to squish it into a circle, but that's always going to happen. So what is this telling me? It's telling me that the receptive field information, and I should say also, this is Nico's presentation will cover this a little bit, but in practice, there's a fairly large amount of geometric information that you see. Can I ask what time I'm supposed to end? 45. Okay, thanks. Okay, quick questions. Okay, great, thank you. Okay, so there's a fairly large amount of geometric information that gets encoded when we do this. If you enrich this data just a little bit, and Nico's presentation will give you an idea how we do that. But the topological information Almost everybody who has ever heard of topology, so if you've never heard of topology before today, maybe you haven't heard this, but you're supposed to be able to deform a coffee cup into a donut. And that was what I was told when I was a graduate student. And I thought, wow, what an incredibly useless thing to be able to do. But we should really think about this as I've got various embeddings of shapes, I've got various versions of shapes, I've got noise, and I want to be able to be insensitive to those things. And so topology only records sort of gross qualitative features, like the fact that that's a circle versus a torus versus a sphere. Things like dimension don't matter at all. You can thicken things up in any dimension, and that's a continuous deformation. So this is a very loose notion of information, but it depends only on overlaps of these intersections of these sets, which is to say only on coactivation of neurons. Okay, so this is information I can definitely get out of the brain, and more importantly, it's information that downstream neurons can definitely access from upstream, right? And we always want to be thinking, what could the brain actually possibly be doing in there, right? And this is something, this kind of information is accessible to neurons natively. So that's really cool because it tells us that maybe these representations are actually the kinds of things you could build a model of neural computation on. So if I start with this notion that this is what what a stimulus space is, or a neural manifold, or whichever of these versions you want to, whatever terminology you want to put on it, I'm allowed to, I can get up to this point without ever leaving the realm of sort of pure math. Okay, but now I should, because we actually want to answer neuroscientific questions, so we want to see this thing in use. So there's this tool called persistent homology. This is what Nico's going to tell us all about. I'm going to give you this one slide that tells you how it works. I start with something, stimuli, spike trains, choose your favorite thing. I compute a pairwise similarity matrix. From that pairwise similarity matrix, I construct some kind of sequence of simplicial complexes, which again, Nico will tell you exactly how we do that. And then I compute the topology in each one of them. And I sew that all together. And I get this thing called the barcode. And the barcode is going to have a bunch of these little pieces. Each one of them is going to correspond to a feature. And the longer a bar is, the more significant a feature is supposed to be. So long bars equal actual circles or spheres or whatever topological thing you're looking at. Short bars probably noise. Although that relies a lot on geometry in order for us to prove anything about. So we will just shuffle our judgments under the rug and pay attention to the long bars, which we know are supposed to be significant objects. Yeah, so all of this to be detailed later. Okay, and so we used this when we got started in order to see if there was geometry using some kind of statistics. But we have, I told you there were questions that I wanted answers to. So here's a question that I think we can answer very effectively using these tools, which is, suppose I have two different stimulus spaces or two different whatevers, and each one of them has a topological feature in it. So here I'm going to imagine a situation where I have, on the left, I have a stimulus I've designed, and this is gratings moving around sort of a gray background, and they can rotate, right? So I've got these pictures. And so I've got some kind of topology on the left that I might be able to detect by similarity between images. On the right, I have some neural activations that I'm recording from somewhere in your brain, right? So on the left, I can do this story I just told you, right? I can say, okay, and the way I'm going to do this actually for this particular picture, sorry, the later one has rotations, is I'm going to keep the direction fixed, and I'm just going to let it move around, but I'm going to let it slide off one side and back on the other, like, asteroids or Pac-Man, or I don't know if there's a modern video game where they do that, but so, right, so I can detect these two circular features using these topological methods straight away. On the right, maybe I do some computations, just right, similarity between spike trains, and I come up with one feature on the right. So now here's a question. Is that one feature on the right either of those other two features at all, right? Is it one of them? Maybe it's just something else the brain is doing, head direction, right? Or maybe it's a combination of the two, or something else, right? So the question is there some formal way I can take detecting these various features and say, these things should be the same, right? Again, building up from the ground up. Similarly, I don't have to have this external thing. I could just have two brain regions I'm recording from, right? I could observe some activity in one set of spike trains. This would be what I would expect from grid cells. Got something over there. Maybe, I don't know, it's hippocampus. Just choose your favorite idea, right? And I just want to know, right? Are those features I've detected, those topological features, the same feature? Is the information in there being represented? Is it the same? So with Iris Yoon and Rob Greist, we built this new math tool that lets us answer this question in sort of the mathematical side. The way we build this, you need more data than just the two abstract things, right? If I just give you two abstract shapes, there's no way to associate them. You need something which can let you go back and forth. And traditionally in topology, what you want is a function going from one to the other. That's almost impossible to come up with in data, because if I knew functions, I would know the underlying spaces. And if I knew the underlying spaces, I wouldn't have to do any of this. So what I can do is measure cross similarity, just like I measured internal similarity, right? I could imagine measuring just a big similarity matrix. Although there's no reason for the cross similarity to be measured in the same way. There might be a time delay, or right, I need cross similarity between images and neural activity. So I've got to come up with some way of saying, I see this neural activity is time locked to this neural activation, right? I need some way to do that abstractly. I take that joint similarity, the cross similarity matrix, right, between the two, and I can construct a new simplicial complex using the same persistent homology kind of tools. It's a little bit different than the one that used for the individual, but what it does is it tells you, when you sew it all together, again, technical details under the rug, happy to take you through them later. It gives you a way to sort of slide a cyclic feature or a topological feature on one side over into the other system. And if you can do that and hit one, then what this does is very robustly tells you, yes, these are the same. It's designed to be very, very careful about that. Great. Mm-hmm. Yep. Yes.
Choosing the lower left.
Right. Yes. Thank you. That's one of my favorite questions to get, because doesn't matter. So there's a wonderful theorem from classical topology, 1950s algebraic topology, called Dowker's theorem, which I have swept under the rug for purposes of this talk. So in order to, I'm actually going to claim that Dowker's theorem is intrinsic to this method, and you need to apply it. So what Dowker's theorem is going to let us do is take one of those two complexes and turn it into the other one, which transforms who my neurons are, or transforms who my vertices are, which lets me actually get away with moving things from one system to the other. And I'll take you all through it afterwards. But like essentially, Dowker's theorem is the absolute key. Oh, it's a question online here. Let's see if I can pull it up. Nope, I cannot. Yeah, if somebody wouldn't.
Superscript stays the same. Is stimulus threshold or just a placeholder represent the excitation?
That's a great question. So in the abstract model, it's just what's above background activity. Sorry, so to repeat the question, is what is the theta, the constant theta represent in the model for the receptive fields? So in the abstract, it's just a placeholder for this is what it means to be super threshold in terms of activity, right? Above background or whatever you're choosing for your model. It can, of course, change from neuron to neuron, just like the Fs can, but you can, of course, sweep that information into one or the other, sort of however you like. So I try to be agnostic about what it means sort of in terms of the neurons themselves, but it's definitely worth discussing. I'd be happy to talk to you more about it, what things we could make it mean. So again, I'm trying to keep my model as sort of agnostic as possible. But yeah, so to Karina's question, Dowker's theorem about which one of those two off diagonals I choose, or Dowker's theorem tells us it doesn't matter which one of those two I choose, and in fact the method needs it. So I need to move back and forth between the two. Um, one is the transpose of the other. Yep. Because I should think about that as one big matrix that is symmetric. Just with the entries potentially computed in different ways. And the first thing you might think to do is, well, why don't I just take the big matrix and do the same process, compute the topology? And the answer is you never see anything. And we have good ideas about why that might be, but it's still sort of in progress.
Is there some sort of topological special source on the what you're talking about, kind of sliding and matching?
Yeah.
Most of what you're talking about, I'm sort of like converting into like linear algebra stats and matrices kind of analog to my head and saying like that. I see the analog of a lot of more familiar data analysis techniques.
Sure.
Matching and so on. But you need some metric of similarity, and sort of that's a little bit over. So you're in a kind of good place in the with the synthesis of complexes or something that, like, you know, you're not, but you can be confident that you're not getting a lot of false positives and seeing something similar.
Yeah. Right. So great. So the question is, you know, what's the topology secret sauce for not having to choose a lot of things like metrics on matrices, things that you might have to do if you weren't using this? And the answer is, these are sort of canonical complexes that you build on the matrices without any reference to anything about the underlying data. And the particular complex that we build, so the real secret is that the topological, these bars are not actually measuring individual elements in a vector space, they are vector spaces. And so there are maps. And the maps between vector spaces are induced by maps on the complexes, which we can construct geometrically. So there's no information going into this except those matrices. That's the only thing we feed in, and what comes out is canonical based on matrices. And the thing that's canonical that comes out in this case is a bunch of maps between vector spaces. And in particular, I should say, because what we have here is the data of a relation and not the data of a function, what we end up with is actually a relation between the barcodes and not a function between the barcodes. But there's a particular choice, which is always going to be there. There's some affineness here. So this paper will take you through all of the gory details, and they are very gory. But also, I'm more than happy to walk you through it. But yeah, no, it's part of the reason to do this is because I'm trying my absolute best to make as few choices as possible. Once I've got that matrix in my hand, I want to do nothing but let the machine do its thing. So now, of course, how you get those matrices is up to a lot of intelligent decision making, right? You have to, those are similarity matrices, so you're gonna have to choose a metric on the spike trains or whatever that is appropriate for this context. Vicky?
Is there any sense that, like, that left on left yellow rectangular matrix could be data that you get from the lab that you then construct the on-diagonal blocks from?
The on-diagonal blocks are going to need to be recorded separately.
Okay.
Just because I don't, right, the cross similarity, there are a lot of different internal similarity matrices which are going to correspond to each one of these cross similarity matrices. Simply, right, if I tell you how neurons respond to images, that doesn't necessarily tell you anything, right? Like some aggregate measure of neurons corresponding to particular images, that doesn't necessarily tell you how individual neuron coactivity is going to look. There's going to be a lot of things that are possible there. So it's just, yeah, it's the sort of complicated nature of similarity measures prevent you from doing any sort of. Now, it might be interesting to know how constrained that is, but I don't think anybody has. That's a very hard sounding question to me. So. Cool. All right. So yes, and again, I apologize for the technical lack of sophistication. We don't have the topological background for me to even write down this algorithm, but it's a cool picture, and I'll show you a picture later if we get there. That might help as well. So one thing I want to say before I do this next bit is, and I want to demonstrate this technique in use, but I need to tell you that there are two ways to demonstrate the same information. This is the barcode that we've been looking at. These are bars, and instead of thinking about bars in terms of intervals, what I can do is I can notice that they are they have two endpoints, and I could just plot that pair of endpoints. And topologists go back and forth wildly between these two things, and they're just different ways of drawing the same thing, because sometimes it's more convenient to draw one, and sometimes it's more convenient to draw the other. So on the next few slides, I'm going to use the one on the right, but you should think of it as just being this barcode. And all I've done now is just taken each one of these bars. So the farther you are away from this diagonal, the longer the bar, the more important the thing. Okay, so here's us trying this thing out. So, okay, so I'm going to build a little network. It's a little feed-forward network, where on the left I've got some sort of stimulus like this, and on the right I've got simple cells. So this thing I described for you earlier in terms of just a nice experiment. But now I'm going to actually make the stimulus a little bit more interesting, because instead of just letting this thing go around and slide off the ends, I'm going to let it turn as well. So there are now actually three circular coordinates, right? There's the torus that these little things live on, and then it's allowed to spin. Okay. So these are not quite biological simple cells. We want them to be a little more topologically interesting. So we've added this funny thing where there are receptive fields that go off this side of your eye and come on this side of your eye. You might imagine that, you're Pac-Man or something. So, okay, so I'm going to make these little stimulus movies. I'm going to play them in there. I'm going to do your standard thing, and I'm going to get these spike trains just by sort of each one of these input neurons is going to have a particular Gabor filter that it likes. Okay. So this is just a little model network, and what I'm going to do now is compute. So this is the persistence diagram, right? So this is the topology of the stimulus. I've got some noise stuff down here because these are actually, right, spike trains out of these Gabor filters. These are, so these are just, because we don't, so I should say this, because we don't have the underlying actual thing, this funny torus, but we've just got points on it, we're going to get some noise, right? We've got holes, so these little holes. But I've got three circular features corresponding to actual, right, to these three directions I just told you about, maybe. I don't actually know, but they're three circular features I compute. And now I'm going to do the other half of this thing, and I'm going to compute the persistence diagram for the simple cells, doing the process I told you, right? I take the correlation matrix, compute this thing. I see three topological features. Cool. And now I'm going to apply this method I just told you about to see whether or not things match. And I get matches between the corresponding colors, so the square and the diamond, the star and the X, and the pentagon and the circle. Those match, and we ran this against a very carefully built null model that captures geometry but not alignment. And what we see is that that we get these very, very good matches between those three features, and with very high probability there are no matches between the other ones. So there is a matching between the input and the output feature, as expected, right? If this didn't work, we should go home. Okay, but you say, look, I probably could have worked this out. This one's the top one. This one's, I could just have drawn some lines, right? I didn't need all of this fancy technology, right? There are three features. There are three features. Those features are obviously the same. So let me add a little more complexity. Now I'm going to build some things that eat outputs from those simple cells, so spike trains produced by the simple cells. And I'm going to design some orientation cells. So those are cells that are only going to be, right, I'm going to train them to only be sensitive to the orientation on the inputs, right? So it's not seeing any of the position information. And then I'm going to train some that are direction cells. So those aren't recording anything about position or orientation. They're recording what direction the thing is moving in. Okay, that's not information that's being represented up here explicitly. It's implicit in the time course up here. Right, so it requires that geometry to exist, but it isn't actually represented as any of these static geometric features. And what I see when I do the same process again is that I do get a match to a feature in the orientation cells. So the orientation was in fact this blue one, because those cells see orientation, and that's the only thing that matches. And there's no match that, right, I get a new feature down here in the direction sensitive cells, which doesn't match to anything upstream because it's new information derived from the old information. So this thing is good at two things. One, explicitly noticing when information is quote unquote the same, right, according to the similarity measure I feed in. and at falsifying things which are not, which is everything you might hope it would do. Hooray. I'm very pleased with it myself. I think it worked out really well. Okay. And then you say, okay, but Chad, these are simulations. So we went out and we got some friends who work on data. This is data from Spencer Levera-Smith's lab at UCSD. UCS, oh, it might be B. I apologize. My D's and my B's. So this is wide field microscopy in mice. So we have neurons recorded in V1 and an anterior lateral area AL, and so these are both early visual areas. So we're going to go ahead and impute some spikes from the calcium time courses. The rat is being shown this very, very simple set. The rodent, it's a mouse, I think, is being shown these very, very simple gradings. There are actually only 4 directions. So this is maybe the simplest circle you can imagine, which makes it actually really hard for these methods to find. But we do some aggregated spike trains. And then without any reference to the input, we just compare activity in the 2 regions. And we're able to match this circle, which we know should exist in V1 because V1 has orientation dependence. But we find the same information being recorded next door. Okay. Well, I think I have run myself out of time before I got to the exciting new stuff. But I am just going to say that I'm just going to give you a philosophy thing and ignore Nico's work entirely. I'm sorry, Nico. Didn't quite get to you. But I stole this from the ICERM workshop, this picture on the right, because it's so beautiful. The ICERM workshop in end of 23 that Karina and folks ran. So what I want to do with all this is the following thing. I want to say, okay, I've got these neural populations spread throughout your brain. Each one of them has this neural manifold. And then what I want to do is, impute that thing. I've got a tool now that tells me when information lines up pairwise. That's pretty cool. So I can compare these populations pairwise and see how information is represented or co-represented or changes, right? But of course, I already, and then I can, glue these things together somehow, right? That's what I'd like to do. But of course, I'm big on doing things that are above pairwise. So what I want to do is fill in this triangle. And we just got a paper out in the archive where we build what we call the multi-way Dowker theorem, which tells you mathematically how this should be done. If you have 3 or 4 or 17 different populations, you can now construct, and then this work is Nico and Iris Vindesilva's 8 people on this paper because it came out of an AIM workshop. And they all did fantastic effort, and I was very privileged to be part of it. But we have a mathematical foundation for starting to ask on the large scale across the entire brain, how does information compare? And I'm very excited for the direction that's going to go in. So I'm going to skip the last 15 minutes of this talk because we are 15 minutes, that would be 15 minutes over. And I'll just say that we want to ask questions about, architecture and about learning. And I will call it there and jump to my last slide because I want to leave some references here to find it. Also, I have to very much thank my sponsors. So Air Force Office of Scientific Research, NSF, for their assistance. We have a couple papers that track or discuss the stuff that I talked about already. And the one thing that I did not get to talk about, which is Nico's fantastic forthcoming work, is that we are able to use these tools in spiking networks to study how topology moves as you learn. And we're able to show pretty robustly that STDP is, maybe as one might expect, but it's really hard to figure out how to show this, is sufficient to induce topology moving sort of forward through networks. So, all right, thank you very much.
So I think what we